Okay, hello, hi everybody, and welcome to the latest in our series of seminars from the EVOLTREE network. This one is going to be focused on genetic and genomic approaches to studying communities, and I'm really pleased to say we've got a great program. 3 seminars coming up over the next month or so. Today, the 30th of November and the 7th of December. So we've got some really good speakers, and I hope we'll have some very interesting discussion following the presentations. We have a slightly different format to the one that we've used previously in our seminar series. We have introduced an opportunity now to have a couple of flash talks just before the main speaker. So today we've got 2 flash talks, which we will hold just before the main presentation. And we'll allow a brief moment of time for 1 to 2 questions following those talks, and then we'll move on to the main event. But if you do have questions for the flash talk speakers, please do hold them and bring them up again at the end of the main presentation. Just before we start, I want to just briefly introduce the EVOLTREE network to those of you who don't know it. So this is an EVOLTREE event, and EVOLTREE is a network of researchers from now 29 institutions across 20 countries. It's been running for more than 10 years, and we're all interested in aspects of evolution in trees and the communities they support. So the network has played a really vital role in bringing everybody together, and currently still supports a series of training courses, a biannual conference. We have calls for small projects and travel grants, and of course this seminar series. All of that can be followed up on at the website, evoltree.eu, or you can follow us on Twitter if you're still a Twitter user, while we're all still using Twitter. But please do follow up, because there's a lot of interesting information there, and opportunities, and chances for projects and travel grants and so on. So please do take a look. There'll be a little bit more information on the next conference, which is coming up very soon at the end of, we'll give you a bit more information at the end of the talks, but do look out for that, because there'll be more detail emerging very soon. So without further ado, I would like to move straight on, and we're going to open with our 2 flash talk speakers. So these will be Jackie Corbin and Hilary Cooper, who are both members of the Cottonwood group in North Arizona University. Jackie's going to speak to us about leaf hyperspectral reflectance and how that can be used to look at variation in plant traits. And Hilary's going to speak about genetic variation of phenotypic plasticity and its impact on tree traits and their associated communities. So can we move straight on to hear from Jackie first? I'll stop sharing and hand over to you.
Okay, great.
That's great.
All right, are you able to see my screen all right?
Yes, that's perfect.
Okay, wonderful. We'll go ahead and get started. Thank you so much for inviting us to speak. We're very excited to share some of our research that we've done in the Cottonwood group, and I'll be sharing some of my doctoral dissertation research on how we can use leaf hyperspectral reflectance to detect genetic variation between Fremont cottonwood trees, and specifically how we can use that to detect disease susceptible and resistant trees and some potential community implications of that. So let me orient you to our study system real quick. This map shows Arizona. It's in the southwestern United States. And each of these black icons are wild source populations of Fremont cottonwood that we took clonal cuttings from. And then we reciprocally planted them into another common garden up in Canyonlands, Utah. It's near Moab, if you're familiar with the Four Corners region. And the base map here shows mean annual temperature So this species occurs at a wide environmental gradient, different elevations, different climates it's adapted to. So we have some hot adapted populations as well as some cooler adapted populations that we've moved up in latitude and elevation to see how they can contend with a novel environment. Now, within this coolest common garden, we noticed that certain genotypes of these trees are either resistant to an endemic fungal species on the landscape, or they're susceptible to it. Some common symptoms of this infestation include clubbing of the tree, cambial splits, so some very severe trunk damage. And if it progresses too much, then it can cause mortality of the central leader itself. So interestingly, we're able to detect these genetic differences between susceptible and resistant trees with leaf reflectance, so how light bounces off of the surface of a leaf. So we can detect the genetics of the tree itself, as well as the disease outbreak and potentially some community consequences. So we did some sequencing on this fungus. We found that it's Cytospora salicina, and with leaf reflectance, we're able to predict which tree has trunk damage due to it with about 70% accuracy. And in tandem with the canker damage that this fungus causes, insect borers also tend to infest these trees, and we're able to detect insect borer damage just with leaf reflectance with over 70% accuracy. So how this relates to community consequences is that both of these infestations tend to correlate with increased occurrence of other fungal species like pleurotus, increased occurrence of different flies and gnats. And then the flies and gnats entice in different avian species such as flickers and sapsuckers who showed increased interest in the susceptible trees. And there's also potentially some interactions between these different taxa in that flickers and flies may be spreading the disease as well, and potentially even other endophytic fungal species. And there may also be an interaction with mule deer. We saw decreased occurrence of mule deer browsing on the resisted trees. Our healthiest, happiest trees tended to be much more foraged upon by mule deer. So some future directions for our group is to see if we can use leaf hyperspectral reflectance to do aerial surveys with UAV drones, flyovers, and potentially satellite imagery. So we're going to see if we can detect this genetic variation in disease resistance and their community consequences with aerial imagery. So I give you just now one example of how we can use leaf reflectance to detect differences in genetically based traits, but there's a wide variety of other traits that we can detect as well. And those include biochemical differences at the leaf level, so differences in, say, structural carbohydrates or defensive chemistry, anatomical and morphological differences in the leaf itself. And then we can indirectly and directly measure genetic differences in functional traits. So things like leaf shape, the pigmentation, but also tree level traits such as biomass. We're able to detect differences in life stage, so whether trees are juvenile or more mature, and where the leaves are in their annual development. We're also able to detect genetic differences in the geography of where the tree is. So whether or not it's adapted to a certain climate, a certain elevation, things like that. And then as in addition to that, differences in hybrid status, sex, community level differences, and of course, pathogens and herbivory. So with that, I'll wrap up my talk and thank you so much.
Perfect, Jackie. Thank you. Really interesting and lots of potential there, I think. Can we move straight on then to the second flash speaker, Hilary?
Yes, hi.
Great, hi.
Let me share my screen. Oops. Can you see that, everybody?
Yeah, if we could just go to the presentation mode. Yeah.
Sure. All right. So thanks very much, guys. I'm going to talk to you today about phenotypic plasticity and how that can affect associated communities of Fremont cottonwoods. And the major goal is to really examine how climate change may alter these community genetics patterns that we've identified. Okay, so briefly about our design again, what Jackie covered earlier. We had 3 common gardens. Jackie just used 1, but this work uses all 3 across a very large environmental gradient. And we had 16 populations cloned and planted reciprocally along this gradient. And this is an example of our central common garden in central Arizona. And then we also did RAD-seq data to get a bunch of SNPs, and we determined that these populations are genetically differentiated. So using replicated common gardens, we can get at phenotypic plasticity, and that is a genotype's ability to change its phenotype in response to environmental change. And so we've done a lot of these reaction norms. This is just bud flush in the spring across the 3 common gardens in the bottom. And so we can calculate plasticity for these different populations. And so a cold adapted population in dark blue here has pretty low plasticity. So here's just the difference between the greatest and lowest value across our environment. So something like 15. And then compared to these hot adapted populations in red show really large plastic responses to this phenology trait. Okay. So I've given you a little background on our system and phenotypic plasticity, and now I want to take you into 2 examples of how this can affect communities. And so first is that foliar arthropods really key into leaf phenology. So in the springtime, when those new leaves emerge, they're very tasty, and so many organisms utilize them for food and habitats such as these galls here. And so what we observed in our hottest common garden in southern Arizona is tremendous variation in herbivory across these populations. And we know that this herbivory is highly tied to phenology. So the earlier these trees leaf out, the more herbivory we observe on them. And then if they flush really late, we have very low incidence of herbivory. And we also know that this trait is highly plastic and it's variable among populations. So again, the hot adapted populations are highly plastic and flush, whereas these cold adapted populations in blue aren't very plastic. And so you can imagine a scenario where these cold adapted populations are experiencing climate warming, and now the opportunity for their growing season has been pushed earlier. And so if the bugs are tracking climate change and maybe flush down here early, even in the winter, these populations aren't matching this early insect emergence. So here we have a phenological mismatch, and we might need to find populations such as this one that is more closely matching timing with their insects to avoid a big phenological mismatch between bugs and their food source. Okay, so moving on to a second example of communities being affected by plasticity. Here, aquatic arthropods really key into leaf litter traits. So when leaves fall into riparian systems, it provides a huge nutrient pulse for many organisms. Aquatic invertebrates and microbes really utilize these leaves, again, for food and habitat. And this is a picture of how we capture the emerging insects to study them. And so we did a leaf decomposition study where we took 6 of these populations across the 3 gardens and collected their litter. And we found that the hot adapted populations in general were fast decomposing leaves, whereas the cold adapted populations, at least in the cool garden, were much slower. Okay, so how will this affect riparian food webs in the future? And so if you look across our environmental gradient across the 3 gardens, we see that hot adapted populations are in general fast decomposers. And so if there's natural selection for these hot adapted populations or artificial selection, if you choose them for restoration stock,
we're going to get fast decomposition across the landscape. Likewise, if these cold adapted populations are experiencing warming, they're going to shift their traits plastically to also have fast decomposition. And then finally, warmer conditions creates less variable litter. So hotter conditions is fast decomposition and less variable, which means there's less opportunity for energy uptake by aquatic invertebrates and a smaller window for food for higher trophic levels, such as birds and fish and bats, and then lower diversity and abundance of organisms. So again, by incorporating plasticity into community genetic studies, we can really test how these genetic community or gene by environment linkages may change as the environment changes. And I've given you 2 examples here to show you the potential. So with that, I'll take any questions and thank you.
Perfect. Thanks very much, Hilary. And thanks to both of you for being bang on time. That was really good.
Thanks. Okay.
So we have an opportunity to just ask a couple of questions now, if anybody would like to. Maybe use if you put a hand up, then we will try to find you in the audience. And I could maybe just ask Jackie, do you think, so it's a sort of, you're detecting with the hyperspectral imagery a kind of sort of inherent biochemical difference among genotypes, is that right? That's present in the leaves but also reflected in the stem?
Absolutely, yeah. It's present in the leaves, and we're also detecting, the really interesting thing about that is that we could detect trunk damage, and also some other studies that we've done have suggested that we can detect root level traits as well, just from the canopy signal. No,
it's interesting, and there's no sense that the leaf biochemistry might vary with conditions independent of the composition of trunk and roots, for example.
Trying to think through that one. So we could detect genetic variation between different environments, if that's what you're getting at, is that there's variation genetically, but also the environment can change the expression of foliar traits quite a bit too.
Yeah, great. Any others? If you want to mull over the talks, and yeah, if you have questions, we can pick up again later. But I think, yeah, thanks again to both of the Flash speakers. They're really interesting talks and adds nicely to the content of the session. So if we could, we will now move on to our main speaker. So he will be Professor Thomas Whitham. So he's a Regents Professor Emeritus at the Department of Biological Sciences in North Arizona University. He is working on community genetics, particularly around restoring threatened poplar riparian communities and pinyon woodlands in the Western US, but is known worldwide really for his work in developing and framing the field of community genetics and genomics, and has a huge impact really through more than 300 peer-reviewed publications now in everything from the top ranked journals on, and even a documentary. So that impact extends not just to academia, but beyond to influencing government level support for using genetics to look at conservation of riparian systems and using forest trees to mitigate climate change. And I hope we'll hear a little bit more about that in his talk. But without further ado, I'd like to hand over to Tom, who will be speaking to us today about community evolution, what it is and why it's important. So Tom, over to you.
Okay, well, good evening, and thanks very much for inviting us. A lot of what I'll talk about today has been summarized in an annual review of ecology, evolution, and systematics article that published in 2020, and I'll add some additional, more recent studies to that today. This first illustration that you see really captures the heart of a lot of what we've done over the last 20 plus years. And basically, the genetics of a foundation tree species then has a major effect on the birds, the mammals, the insects, the fungi, the soil decomposers. And those organisms in turn can feed back to affect the performance of the tree. And so this leads us into community evolution. And so I want to talk about what is it and why is it so important. And this next slide shows you some of the habitats that we study here in the American Southwest. And so this shows a riparian corridor surrounded by a very arid landscape of pinyon pine and juniper. And we've studied both of these systems of pine and cottonwoods throughout the Southwest. And so here I'd like to start by defining community evolution. And it is the outcome of selection operating at multiple levels, individual, group, community, that results in the differential survival and proliferation of communities over time. And today I'll concentrate most on individual and community level selection and evolution. And so community evolution is really based upon multi-level selection. And multi-level selection is a controversial issue, at least for some groups. And Williams in 1966 asserted that selection acts most effectively, if not exclusively, at the individual level. And he argued that, I quote, in explaining adaptation, one should assume the adequacy of the simplest form of natural selection, that of alternative alleles in Mendelian populations, unless the evidence clearly shows that this theory does not suffice. And we feel it does not suffice. And so this has been widely accepted, especially by ecologists and behaviorists. And so there are numerous problems in relying only on Mendelian genetics and individual selection. And so the first thing is that the theory does not suffice in non-Mendelian populations. And so this would include multiple alleles, codominance, epistasis, pleiotropy, sex-linked traits, polygenic traits, community interactions, environmental interactions, and clonal organisms. And these things are so common that we feel that people who look at individual selection alone are missing literally a great deal of the action, the evolutionary action. And so if you don't look for it, you'll never find it. And we think people really need to start looking at these other levels of selection of group and community level selection. And so the second major thing is that increasingly we're starting to find out that the individual is a conceptual fallacy, because most plants and animals intimately interact with communities of symbiotic organisms that are essential for their survival. And so Gilbert et al. Discusses this issue. And so we're dealing with the holobiont, and that is defined as an assemblage of the host and the many other species living in or around it, which together form a discrete ecological unit that selection can act upon. And then number three, this view ignores higher levels of selection, again, such as group and community level selection. And then fourth, most or all species live in a community context. And so it's just far more realistic. And so we need to take it into account. Okay, so over the last 20 years or so, our group has published well over 130 papers on the genetic variation and population and community traits that are central to the holobiont and beyond. And so I don't have time to go into this slide, but I just want to emphasize that there's a lot of studies coming at this from many different directions, and they're all focused, or most of these studies are focused at the holobiont level of individual tree genotypes and the communities of organisms above and below ground that they interact with. And so the holobiont for us has revealed a lot of important genetic information that is important in the evolutionary process. Okay, so here I list three major criteria or requirements that are absolutely necessary to support the hypothesis of community evolution. And the first one is that intraspecific genetic-based variation in community phenotypes. Okay, so we need to demonstrate that. Then we need to show that these community phenotypes are heritable, and then we need to be able to show that differential survival and reproduction of community phenotypes. And so these are three criteria. And so if you're going to support the hypothesis of community evolution, all of these three things must be demonstrated. And you can negate this hypothesis by showing that one or more of these are not present in your system. I also want to emphasize that these are equivalent to requirements for evolution at other levels that have been published for many years. Okay, so I'm going to go through each of these requirements and present you with specific examples of what we found. And so here I'm going to give several examples of genetics-based variation among communities' phenotypes that are greatly magnified by species interactions. I can't emphasize enough just how important species interactions are. And so the existence of variation, genetics-based variation in species and community phenotypes is driven largely by individual and population traditional phenotypes, such as leaf morphology, water use efficiency, phenology, architecture, bark thickness. And so all of these are, in our system at least, are genetically based. And the combination of all of these traits through sexual reproduction and assortment results in all combinations and permutations among individuals within a population, so that some have a few or all of these varying levels. And so because all of these traits are really important to individual species and whole communities of associated species, that results in community phenotypes that are unique to individual tree genotypes. And the same thing goes for ecosystem traits such as trophic structure, biodiversity, community stability, and so on. And so this is the driver of a lot of what we see at the community level. And so there should be a link between these traits and the community phenotypes and the ecosystem phenotypes. Okay, so that's what should be driving it. And so now I want to give you just a few examples of this. And so an example, my first example is with the interaction of two foundation species. And so the genetics-based interactions of two foundation species, in this case, cottonwoods and a pathogen, define a much larger arthropod community. And so here we have in the upper left-hand corner a leaf pathogen. It's very common in the Western United States. And then this shows a common garden in which you can see this tree is pretty much clobbered by it, whereas the tree right behind it or over here are genetically resistant to this pathogen, and it's not having much effect on them. And so we know there's a strong heritable variation in resistance to this pathogen. And so then we wanted to quantify experimentally how the interactions of the pathogen and the tree would affect the arthropod community. And so then we performed an experiment in which we cloned trees from the wild and which were represented in the common garden. And half of them we inoculated with the pathogen, and the other half remained as controls. And then here shows the plants in pots. And so here we have a common garden within a common garden. Okay, and so the next slide shows you our basic findings. And this is a non-metric multi-dimensional scaling ordination. And so each dot or symbol represents the arthropod community found on treatment and controls for narrowleaf cottonwood, Fremont cottonwood, and their naturally occurring hybrids. And so here with Fremont cottonwood, you see the arthropod community on the pathogen inoculated plants is very different than the control plants. And the same holds true for narrowleaf cottonwood, in which the arthropod community on pathogen and control plants is very different. And then with the F1 hybrids, even greater differences between the pathogen and the controls. And these differences are all significant. And so what this is showing you is a plant genetics by fungal pathogen interaction that creates more community phenotypes that selection can act upon. And so how much genetic variation do we have in community phenotypes is going to be very important in terms of selection and evolution. Okay, so my next example is looking at genotypic variation in a foundation tree, how it generates different ecological interaction networks that selection can act upon. So in this study by Matt Lau, we have, again, a common garden experiment in which we have 10 different tree genotypes shown on the left, and on the right, we have 44 different arthropod species that are commonly associated with this tree. And so then the connecting lines between the two, the broader the line, the stronger the interaction, the narrower the line, the weaker the interaction. And so what you can see here is that different tree genotypes have very different interaction networks. And so tree 1017 has some strong interactions with many community members, whereas tree 1020 has much weaker interactions with all of these community members. And so this is generating, the interaction networks are generating a lot of variation that can select upon either the tree itself or the community. Okay, so now another example, moving this up to whole communities, and a study published by Lamet in the Journal of Ecology. We're going to examine community genetic correlations. And community genetic correlations are defined as changes in the composition of one community among plant genotypes that are mirrored by the changes in the composition of another community. And so here we have very different communities above and below ground, lichens, leaf pathogens, arthropods, twig endophytes, soil fungi, and soil bacteria. And frankly, when we started this, we thought this was a long shot. Why would these be connected? But in the network analysis, we looked at these community interaction networks, and we found very strong correlations between these communities that were based upon individual tree genotypes. And so if you, all of these, so the communities, my next topic will be on heritability, and all these communities are heritable. But so that means that if you change the genotype of a tree, you will change the community of arthropods living on it. But not only that, if you change the genotype of the tree, not only will the arthropod community change, the endomycorrhizal fungi will change, the lichens on the trunk will change, the fungal leaf pathogens will change, the twig endophytes will change, the soil fungi will change, and the soil bacteria will change. And these connecting lines are all highly significant. So this was a level of connectivity that kind of blew us away, but it suggests that organisms are far more interactive and connected than we initially dreamed of. And so this was a wondrous moment, at least for us. Okay, my next example is with community plasticity that is reflected in community reaction norms for species richness and abundance. And they're both saying the same thing, so I don't need to repeat explanation of both graphs. But this involves an analysis of 250 arthropod species found on pinyon pine. And there were 3 types of pinyon pine included here. The blue line shows scale-resistant trees or scale-resistant genotypes. The brown lines show scale susceptible genotypes, and the green line shows genetically susceptible trees in which the scale has been removed, experimentally removed for 15 years. And so this is all, these trees are naturally occurring in the wild, all growing intermixed. And then on top of this, we include climate change because in 2002, we had a record drought year. And then in 2003, we had a normal year. And so these arthropods were quantified on individual trees over this 2-year period. And this represents a paper published by Stone in Frontiers in Plant Science in 2018. And so what you see here are the reaction norms or plasticity are genotype specific. And so in the susceptible trees, it's basically flat line. In a drought year, they don't support many species, hardly any, in fact. And then in a normal year, they don't support much either. And so then we find steep reaction norms for genetically resistant trees that are shown by these blue dashed lines. And in a record drought year, they're not supporting much either, but a lot more than the susceptible trees. But then in a normal year, they show very steep reaction norms where they're supporting a whole lot more. And so this reflects just how important stress is in negatively affecting biodiversity. And then in the scale excluded trees, even though they're susceptible, when you take the scale insect away, they behave like resistant trees. And so this shows that the scale is driving the interaction, and the presence or absence of the scale is defined by the resistance traits of the tree, and it is influenced by environmental stress or climate change. And so basically, what we're showing is a scale insect by tree genotype interaction with the environment, drought, to affect arthropod communities. And this is important because each of these genotypes is creating different community phenotypes that selection can act upon. Okay, so I've given you several examples of variation in community phenotypes, which was the first criteria. And this is, we have many examples of this. And so these are just a couple from all of those studies that we published demonstrating variation in community phenotypes. That's genetically based. Okay, so the second criteria that we must demonstrate is that these community phenotypes must in fact be heritable. And so heritability here is basically defined as the tendency of related individuals to support the same community and ecosystem processes. Okay, so here's an example of heritability that you can see visually. And this is not at the community level, but this is looking at a traditional phenotype, say, of growth. And it really pops out here because this is one of our few gardens that was set up in which a block design in which we had 20 or more clonal replicates of each tree genotype. And so here you can see genotype number 1 is growing very well, rapid growth rate, and a different phenology than genotype 2. And genotype 2 is also doing very well, but it's growing at a fraction of the rate of genotype number 1. And then genotype number 4 completely died out, and genotype 3 is kind of intermediate between 2 and 4. Okay, so this kind of variation in growth that's genetically based is important to many community members that really key in on growth rates and phenology. And so it's not a stretch of the imagination then to say we should start looking, when we have these kind of obvious traits, should be looking at the community above and below ground. And that's what I'll do in some of my next slides. And so here is community heritability. And this was the first example of community heritability that we published in 2006 in Evolution, paper by Steve Schuster. And again, this is a common garden study, and each dot represents an individual tree and the community of arthropods found on that tree. And so trees with this that are clonal replicates of the same genotype all have the same symbol. And so here we have 4 clonal replicates of the same tree genotype and their respective communities. And you can see they're all grouped together. Here we have 4 clonal replicates of another tree genotype in the blue squares, and they also group together in their arthropod communities. And this is important to note here that they're randomly planted within a common garden. So it's not because they're in a block all together, they're randomly planted with all the other tree genotypes. And so by looking at the within and among group variation, we can quantify community heritability. And in this case, it was like 0.60, meaning about 60% of the variation in the communities can be accounted for by knowing the genotypes of the individual trees. And so this was the first example. And now we've published since then many more examples. Here's a paper by Busby on the leaf pathogen community, another paper by Jen Schweitzer on the soil microbial community, another paper by Jamie Lammett on the twig endophyte community, and another one by Lammett on the lichen community. And the heritabilities of these different community types range from 0.20 to 0.70. So in very diverse organisms, we're finding community heritability. Okay, the next slide comes back to interaction networks. And interaction networks, or the interactions among species found on individual trees, amazingly is also a heritable trait. And so here we have 3 different genotypes of trees, and the interaction networks on those three. And you can see they look very different, and they are statistically quite different. And this is unpublished data by Matt Lau. And then here they are all together, the combined interaction networks. And then here we look at an ordination of all the different tree genotypes that were replicated, again, in a common garden. And so here we have the mean interaction network for an individual tree genotype and its clonal replicates, which gives us the confidence limits around that tree genotype. And so here we looked at the heritability of different network metrics. And so network similarity has a heritability of 0.41, the number of links in the network, a heritability of 0.32, and the evenness of the interactions, heritability of 0.33. And so not only are the species and richness of those species on a tree genotype heritable, but the way they interact with one another is heritable. And that, again, was a really important revelation to realize that even the interactions themselves could be quantified as being heritable traits. Okay, so now I want to talk about the heritability of community stability, which is an ecosystem trait. And this is a study by Keith et al. With two studies combined here, one in ecology and the latest one in Proceedings of the Royal Society B in 2017. And this is a common garden study involving 133 arthropod species. And basically, we have these different tree genotypes, and community stability is measured as the turnover rates from 1 year to the next year to a third year. Okay, so basically what he did is quantify the arthropod community on trees in a common garden that were randomly planted from over a 3-year period. And then he looked at the turnover rates from 1 year to the next, to the third. And so if there's a high turnover rate, that would be a low stability community. And if there's a low turnover rate across those 3 years, that would be a much more stable community. And so like tree genotype 1008 here had high turnover rates and wasn't very stable. And here we have tree genotype 1000 has a much lower turnover rate. And you can see the confidence limits around that and the statistical separation of these different communities. And basically, community stability was heritable. And let's see, the heritability was 0.32. So about 32% of the variation in community stability is genetically based upon individual tree genotypes. Associated with that was also heritability of species richness and abundance. So biodiversity and stability are all heritable traits. And if I'm doing a restoration project and I plant genotype 1008, I can destabilize the associated community. And if I plant tree 1000, I can stabilize that community. And so this again was an important finding that really needs to be taken into account in terms of restoration. Okay, so I've given you some really diverse examples of heritability. And so I think we've really supported the first two criteria of community evolution. And here, I'll move into the third criteria of differential survival and reproduction of community phenotypes. If that occurs, that results in community evolution. And so here again is a case where climate change is really important to understand, because here we're moving into an area where we're starting to document that climate change is not only an ecological event, it's actually an evolutionary event, because the effects of climate change are not random, they are genotype dependent. Okay, so here's a hillside composed of two dominant tree species. Most of the dead ones are juniper or pinyons, and most of the green ones are juniper. And so on this hillside, almost all of the pinyons have died, but there are 4 pinyons that survived that are circled here. And so these surviving pinyons, we now know, are genetically different than the trees that died. And so the trees that died because of community heritability, we know that not only did the tree die, but the community that it supported died. Whereas the trees that survived, not only did they survive, but their communities survived. And so now we're in a situation where you can start to visually see how community evolution is occurring, because this would result in a change in community phenotype over time. Okay, so looking at the community level now, this photograph shows you Kitty Gearing, who has pioneered a lot of the mycorrhizal fungal mutualist studies in our group. And here we have a common garden with about 2,500 trees of pinyon pine. And here we have a seedling from a drought intolerant mother. And here we have a seedling from a drought tolerant mother. And you can see they're very different in size. And this is the general pattern. The drought tolerant seedlings grow faster and bigger than the drought intolerant seedlings. Okay, and so here what she has done is to look at the mycorrhizal feedbacks. Well, she's looked at the mycorrhizal communities on these individual trees and the feedbacks that affect pinyon performance and community evolution. Now, these feedbacks are important for evolution, but not necessarily a requirement for community evolution. They can certainly speed it up, but it's not a requirement, at least according to these published studies and others that have emerged. Okay, so this next slide shows you a paper in our review article, and this is based upon several studies published in PNAS, Global Change Biology, and Molecular Ecology. And these panels demonstrate all the 3 criteria that I set out at the beginning. And so in the upper left-hand panel, we see variation and heritability of community phenotypes in the narrow sense. So we're not dealing with clones here, we're dealing with mothers in the wild and their seedlings. And so here, the large circles represent the mycorrhizal community found on drought-tolerant mothers in the wild, and the brown symbols here represent the mycorrhizal community on drought-intolerant mothers in the wild. And you can see that they're really different. And then if we look at their seedlings in a common garden, we see that the seedlings, well, this is in the greenhouse, and the seedlings were inoculated with the mycorrhizae that we find in the wild for both drought-tolerant and drought-intolerant mothers. So they got everything that would occur in the field. And so what you see, though, is that even though they were inoculated with everything, the seedlings only got acquired the mycorrhizae of their mothers. And that goes for both cases. So the mycorrhizae here are being quite specific. And since the seedlings are just like their mothers, this shows narrow sense heritability of the mycorrhizal community. So in this one slide, we're seeing the first 2 criteria fulfilled. And so now I want to look at selection that occurred during a record drought, shown in panel C. So in panel C, we see what the frequency of the different genotypes of drought tolerant and intolerant trees on the landscape were prior to the drought. And you can see here that prior to the drought, drought intolerant mothers were about 75% of the population, whereas drought tolerant mothers were obviously much less. Okay, so then the record drought occurred in 2002, and literally within just 1 year, the frequency of drought tolerant and intolerant mothers changed on the landscape to be about 50-50. And so this is then clearly evidence of evolution at the tree level, the individual level. But this then raises the question, when we're looking at evolution and we look at the individual, is it the individual tree and its genome that's being selected for or against, or is it the mycorrhizal mutualist community that are mutualists that's being selected for or against, or is it both? And so most people don't ever take this into account. And so we really need to merge these kinds of studies. And in the upper right-hand panel, we start to look at how important the mycorrhizal community is. And so this again is a greenhouse experiment that looks at seedling dry mass from both drought tolerant and intolerant mothers and their seedlings. And so here we inoculated the seedlings from drought intolerant mothers with their natal mycorrhizae, and then we have them growing in sterile soil, which allows us to see how much benefit they get by having mycorrhizae. And then in this third area, we have the trees were seedlings were inoculated with the other mycorrhizal community of drought tolerant mothers. And again, these mycorrhizae are so specific, they would not even form, they were not able to form a association when they received the wrong mycorrhizal types. So that was pretty interesting. But then we also see that if you have mycorrhizae, you get a big bump in performance, but the bump almost a doubling. And then for the drought intolerant or drought tolerant seedlings, they got an even larger bump by having mycorrhizae, a 2.5-fold increase. And so, and that increase is much greater than what the intolerant mothers seedlings got when they were inoculated with their mycorrhizae. And so basically this is saying having the right mycorrhizae gives you a much better growth rate. And so then the sterilized soil basically looks at the genetic-based differences of the tree alone, and there's not much of a difference. And so you might make the argument that it's not about the tree genetics, it's actually more about the mycorrhizal community, which was a bit of a revelation for us again. And so we're always left with this thing. If you don't look at the whole community, the holobiont, you may be missing the real understanding of what's going on. And so the community context becomes incredibly important. Okay, so then then can we actually see community evolution occurring at the community level? And so these studies have been going on for a long time. And so we have a 16-year period in which we know the communities of drought intolerant and drought tolerant mothers and their associated mycorrhizal communities. And so here, 1994 was before the record drought. The record drought kicked in about 2002. And then we have the mycorrhizal communities after the record drought, and the record drought is still continuing. And so we're in a mega drought. And so what is showing here, as on the drought intolerant mothers, the mycorrhizal community is changing on the landscape and starting to merge or become more similar to the drought tolerant mycorrhizal communities. This then is a change in community phenotype over time, which fulfills the third condition. And the main way it's changing over time is through the differential mortality of trees and their associated mycorrhizal communities. And so by the time we get to 2010, a fraction of that community and those tree genotypes is left. And then you can see it's very different for the drought tolerant trees. Their communities haven't changed much because they are basically highly adapted to greater drought conditions. And so I think with this example, we have fulfilled all three requirements. Okay, so I want to emphasize that the community is very large. And so I've given you the mycorrhizal community and the tree, but here is a summary in another study that looked at about 1,000 species found on these trees. And so here the one line means that the abundance of these various groups is the same on drought intolerant and drought tolerant mothers. Anything that deviates from equality and has an asterisk on it is a significant deviation. And so here you can see the decomposers, the EM fungi, the vertebrates, birds and mammals are all significantly different, in which some are greater on drought intolerant, some are greater on drought tolerant, and only one group, the heterotrophic bacteria, did not show a significant difference. So the community is large, and basically it generally gets ignored. And we're so focused on the individual, we miss the community effects. Okay, another example here is with introduced species, Rocky Mountain elk. It is not native to the American Southwest, and so it's an exotic to our region. And then it feeds on many plants that are naive to elk browsing, and one of them is this goldenrod. And so the U.S. Forest Service has established elk exclosures because there are a lot of plants that are naive to elk browsing, including aspen. And here is one of those elk exclosures. And so inside the elk exclosure, we see a goldenrod that is flowering and normal. And just outside the elk exclosure, we have another goldenrod that has been browsed upon down to the ground. And one thing that we, in the wild, in these exclosures and outside, we found that observationally that the plants outside of the exclosure flowered about 3 weeks earlier than the plants inside of the exclosure. And the best explanation we have for this difference is that elk has selected for plants that flower earlier, because by flowering earlier, they can largely escape the effects of elk browsing in terms of their reproduction. And then we found that they had very different arthropod communities associated with both groups. And so this could be genetically based, it could be environmentally based, or both. But this would have to be micro side effects if it's environmentally, because they're growing pretty damn close to one another. And so the main way to get at this is to, again, create a common garden. And then, so we collected seeds from inside and outside of the exclosures, from multiple exclosures around northern Arizona. And then we planted them in a common garden where they were, none of the plants were exposed to elk herbivory. So they had the same environment of no elk herbivory. And so then what we found was that indeed it's genetically based, because those derived from elk browse populations flowered about 3 weeks earlier than those derived from no elk populations. And so they flower about 3 weeks earlier when they have a history of elk exposure. So there appears to be a genetic-based difference there. And then if we look at the arthropod communities, again, they are very different. Here we have the arthropod communities from elk excluded populations. They're all different than the elk browse populations, arthropod communities that you see here. And this is involving 45 different arthropod species from very different arthropod orders. And this is a study by Smith et al. Published in Ecology. Okay, so I think we've met these three criteria. And so now I briefly need to get into two other categories. And I want to move beyond community evolution at the holobiont level, where we scale up multi-level selection to not just individual plants, but communities of plants. And I won't talk about group selection, but this is ponderosa pine, and you can see here a group that survived, here is a group that didn't survive. And so everything I've presented you so far has been on the individual in this holobiont, but I think group selection here could become very important. And generally when I find things like this, if it looks different, it generally is different genetically. And then to the community level, which is what I want to present a couple of examples on here. And here at the community level, I'm talking about the tree, the shrubs, and the understory forbs and grasses. And so we need to scale up to even larger scales of multi-level selection. And so here's an example that I'm really excited about, because this is scaling up between different ecosystem types. Here we have a terrestrial habitat type, and then here we have an aquatic ecosystem. And cottonwoods are riparian trees that create gallery forests, and the genetic footprint of a tree extends beyond the tree itself into an adjacent aquatic ecosystem. About 70% of the nutrients in this aquatic ecosystem come from the leaves, the falling leaves from this terrestrial ecosystem. And so what Zacchaeus Thompson did for his PhD work was to create insect or macroinvertebrate community emergence traps. And the leaf litter at the bottom of the stream was removed and replaced with individual tree genotypes of leaves from a common garden where they've been grown under the same conditions. And so each of these different traps has a different genotype of cottonwood leaf litter. And he then looked at the arthropod macroinvertebrates that were emerging after having fed on these different tree genotype litters. And the next slide shows you each dot then is a different tree genotype of leaf litter and the community of macroinvertebrates that emerged with the associated confidence limits. Here's a different tree genotype litter and others. And basically what this is showing is that different genotypes of cottonwood leaf litter support different stream macroinvertebrates, and this too is heritable. That accounts for, I think, about 25 to 30% of the variation is accounted for just by knowing the genotype of the tree. So here we are scaling up to looking at the genetic footprints at higher scales. Okay, one last example on this is a large restoration experiment where we made a restoration experiment we made a restoration project an experiment by planting 17,000 trees at the Cibola National Wildlife Refuge. And there were 3 different species of riparian trees in this, and there were 1 species of cottonwood and 2 species of willow, and their associated arthropod communities. And so in this planting site, as you can see right here, this is what it looked like 18 months after planting. Okay, so the experimental design here was all 3 species were planted in blocks in which they came from the same sites and were growing adjacent to one another and had an evolutionary history of association of living together. And then we created other blocks using the same tree genotypes, but they were mixed and matched across sites such that they were experiencing each other for the first time. And so the ones that had come from the same sites and with the same history, we referred to them as sympatric blocks, and the ones that were mixed and matched we referred to as allopatric blocks. And then the next slide shows you the productivity, the annual net primary productivity of the 3 different species and in these different blocks. And in each case, here's the combined effects of all the blocks from different sites. And so it's showing you that the productivity on average in the sympatric blocks is on average significantly greater than the allopatric blocks in the light bars. And overall, it's about a 20% bump by having plantings in which we have intact communities that have an evolutionary history of living together. And so this is evidence of adaptation with one another, apparently to minimize competitive interactions. And so sympatric blocks do better than allopatric blocks. And restoration hardly ever uses intact communities because, at least in the United States, they tend to come from, they're never matched as intact communities. And so I think this is an important way of getting better restoration efforts, and it demonstrates that the genotypes of your neighbors really matter. Okay, and this also affects biodiversity and the arthropod community. If we look at the sympatric plants and the allopatric plants, in both arthropod richness and abundance, they're both significantly higher by a large degree in the sympatric plantings. And so this is another way of getting higher biodiversity. Okay, so this takes me to my final point. And this is, we expect that global change is generally an evolutionary event that will fundamentally alter community trajectories and the extinction of whole communities on the landscape. Okay, so this is not just an Arizona or Southwest United States problem. This, I think, is a global problem. And so we need funding to develop genetic solutions which are rare in the United States. And so here's an example of what's happening in the US with a riparian system. This is the Bill Williams River and the Bill Williams National Wildlife Refuge. And this was considered one of the 10 most pristine rivers before the droughts started to really nail the American Southwest. And so what you see here, we know that different tree genotypes support different communities, and these do not clone naturally. And so what these dead trees, those tree genotypes and their communities have died out. And here we have maybe the future of restoration as the few trees that are surviving here, these tree genotypes and their communities are surviving. Now, we don't actually have the data to say that this is genetically different than this. And so that's a major emphasis of future research. This shows you what it looks like in the understory. I mean, it's pretty devastating. And this is also especially important because there are 30 sensitive, listed, and endangered species associated with this river system. And so we think we're going to see some really bad effects of climate change here. And all of this is associated with record droughts, and the record drought is now becoming the norm. The record drought first started to be really kick in the year 2000, and it's been that way ever since. The American Southwest is an ongoing mega drought that began in 2000, and Williams, in a 2020 Science article using tree ring studies, demonstrated that it was the second worst drought in 1,200 years. And it's projected to continue on indefinitely. Okay, so what we're proposing is to plant a network of reciprocal common gardens across the American West. So we want to put common gardens all the way here in these border states with Mexico, the intermediate states, and all of these higher latitude states bordering Canada. And so we think that with these kinds of common garden studies, we can come up with identify the stocks that can survive current and future climate change conditions, because there will be reciprocal common gardens. Okay, so we're working with our US congressional representatives to fund a national network of these reciprocal common gardens. And so we now have a bill pending in the US Congress, House Bill 5145. And if that's accepted, who knows? I mean, this is politics. But the goal is to obtain funding for $130 million to set up these common gardens and research into riparian forest, community genetics, and climate adaptation. And we feel that community genetics and genomics can play a major role in developing genetics-based solutions. And so I'm kind of hoping that this should be and will be a global effort. And hopefully our colleagues in Europe would be interested in this, because then across the globe, you could really do comparative studies and merge our findings on an even much larger scale. Okay, so I'll end here by acknowledging our great group of collaborators in community genetics and genetics-based restoration. The skills, these are our current collaborators. It would be much larger over the 25-year period, but their skill sets range from genes to ecosystems and microbes to vertebrates, basic, applied, land managers. We've been continuously funded by the National Science Foundation. These represent some of the institutions that have also been involved with this. It includes our European colleagues as well, and Canadian and Australian. And these are the land management groups that have donated land for long-term 50-year plus common gardens in the United States, in which they buy into this because the common gardens, findings from common gardens can then be related to their adjacent lands and better management practices. And I'll end there, and I think we're very excited about the long-term potential of a community genetics approach. And so thank you very much, and I'd be happy to take any questions.
That's fantastic. Thank you, Tom. I'm sure really impressive work, and I'm sure there'll be lots of questions, and I'm sure there'll be lots of people interested in the potential for collaboration if the work can be extended around the world. So can I just open this now for everybody? If anybody would like to ask a question, can I ask you to just use the raise hand icon, and it could be a question either for Tom himself or for either of our flash speakers at the beginning. I'll just open the floor. Okay, would anybody like to go first, or shall I kick off? Felix, is that a hand, or are you just adjusting? Okay, Kata, I think you have a hand up.
Yes.
Please.
Thank you very much for your great talk. This was really, really inspiring. And I have a question. So, I mean, you showed these really big differences in communities between the different genotypes, for example, like genotypes that are drought tolerant or drought susceptible, but we are already struggling in identifying these genotypes or phenotypes that are clearly drought tolerant or drought susceptible. So I was wondering, how did you know in the first place? Did you just go out in the field and spot the trees that were doing better or worse, or which was the first step to differentiate the two groups of tolerant and susceptible drought phenotypes, for example, in the trees?
Well, I spend a lot of time in the field, and I look for I often look for phenotypic variation. And when I find it, especially between plants growing adjacent to one another, you never know, is it genetically based, or is it a difference in microenvironments? And I grew up in a wholesale nursery in which several generations of family members made their living by identifying genetic variants that could be patented for commercial purposes. And so it's kind of in my blood. And once you get keyed in on looking at genetic phenotypic variation in the field, you can test it by common gardens. And so a nursery is basically a common garden. And so automatically you would say, well, let's test it and see if the trait is stable. And if it's stable, then you can patent it. But then in my, for over 40 years, I've taught a field ecology course, which the students call commando ecology, because we just go out into the field and we look for pattern. And in one such field trip, the pattern was we saw a difference in architecture. And so those pinyon pine you saw had a different architectural type, one a more shrub-like architecture and the one a more upright architecture. And that architecture was related to insect resistance. But it's also related to drought tolerance. And interestingly, the insect resistance trees are the most, the insect susceptible trees are also the most drought tolerant trees. Mm-hmm. Okay, so that's exactly the opposite of what you might expect. So resistance to insects is associated with drought tolerance. And so we think it's a balanced polymorphism. So under one set of conditions, this set of trees does best, and under another set of environmental conditions, these other trees does best. And so that a lot of students that were involved in the class then continued work on this for their master's and PhD degrees. And so it just, once you have a common garden, then people in very different disciplines can then plug into it and start working on it, whether you are working on molecular genetics or whether you're dealing with mycorrhizae or insects or pathogens. You can all work on the same trees in a common garden. And then, amazingly, you can start to merge all of your skill sets because you've all worked on the same trees, and then you can combine your data. And so here at this stage of my career, I'm really interested in trying to do the synthesis and merge, like 40 years worth of efforts and pass these common gardens on to future generations. Because we're dealing with long-lived trees, and we really need to make sure that these are leveraged into the future. And so that's basically how it happens.
Can I pop in a second?
Thanks a lot.
And I just wanted to add something on the cottonwood side to that question. And so the pinyon system, there's this really clear architecture trait that you can key in on. And cottonwoods, we haven't quite got that as well, especially now that drought is so strong that it might just mask any sort of phenotypic trait that you can see in everything is dying. And so the pictures that Tom showed on the Bill Williams, we actually went out and collected as many leaves from trees that were in the process of dying. And so that, I think, for a lot of systems, you just have to go before sort of these big climate change stressors to sample everything, and then maybe during, and then after, to really see, okay, what were the genetic basis of survival for, so you have to do it kind of proactively instead of finding a trait and being able to put it in a common garden if there isn't a super obvious phenotypic difference in some cases.
Yeah, I would just add to that, in the cottonwood system, I did my PhD work on the cottonwoods, and there are some visual differences there as well, because I noticed that some trees just were loaded with gall-forming insects, and an adjacent tree had almost none. And that was my first foray into the community side, because I saw these differences in the field, and I didn't know if it was due to genetics or environment. And so we established our first common garden in 1982 to actually quantify was it resistance or susceptibility. And so then if you have a common garden going and you start your studies earlier in your career, fortuitously for this research, it just happened that we went from far more normal climatic conditions, and then climate change really kicked in the American West. And so we had data before and data after. But at this point in time, basically, we have to go out. If you don't see differences, either in insect communities or architecture or any of these other phenotypes, what we need to do are big samplings of trees, and then look at them, create common gardens and expose them to different stressors, and then quantify that. And then what Jackie talked about with the hyperspectral imaging, there are numerous papers, well, I shouldn't say numerous, there are a few papers that argue that the hyperspectral imaging is an equivalent to whole genome sequences, because you can, with hyperspectral images, you're getting thousands of spectra. And so you're getting up to thousands of traits simultaneously. And this has the potential to really revolutionize, because potentially at a single time in using handheld equipment or a drone or a flyover. I think the potential is that we can really potentially in the long term phenotype whole forests, millions of trees for hundreds of traits simultaneously at one point in time. And then as those die off or drought really kicks in, we can look at who lived and who died. And so we really need a lot more of these kinds of studies. And I think we can really up our data accumulation by using this hyperspectral techniques.
Thanks, Tom. Trevor, you have a hand up.
Hello there. Can you hear me? Oh.
Hi, Trevor, we lost you.
You're on mute.
Yes, you're muted, Trevor. Yeah.
It's not unmuting it, old bugger. How do I undo this?
We can hear you now.
Oh, you can hear me now, can you? Okay, good. Hi, Tom. Yes, Trevor Spedding here. I just wanted to pick up on a comment that Katharina made there. It occurred to me it might be harder to see these phenomena in some European forests because, of course, they've been interfered with or manipulated by humans for, in some cases, thousands of years, with people planting species that they like, removing species they don't like, moving stuff around. Whereas you've probably got more pristine forests out your way than we have.
What's the question?
Did you catch that? It was just whether or not, do you think it would be harder to see that sort of the phenomena in European forests because the state of a sort of human interference with European forests might be greater than it is in the US?
No, I think it would be equally productive. I think, so in the US, we're actually proposing putting common gardens in cities because our cities with the heat island effect are anywhere from 5 degrees to, well, let's see, I'm trying to remember whether it's in Fahrenheit or centigrade, but
Especially at night.
they're much hotter than adjacent countrysides. And so the urban and the wildland differences are really great. And so what we're proposing is along our freeways or often there are huge plantings in which, often it's exotics, but it should become experiments of the city landscape. And so the nurseries that generate literally millions of dollars worth of billions of dollars in income are always outside of cities where the land is cheaper. And so they're producing stock that is not often not adapted to the urban landscape. And so by having these common gardens in hotter environments in cities, we can actually identify an effect of climate change where it's been ramped up and even greater than what we see in wildlands. And so it's a great research opportunity. And I think we should avail ourselves of it because I think we have a lot to learn. And we have the land in the cities. If we can just talk the cities to actually use native stock, which they should be doing anyway. And then when we have outbreaks of pests like the emerald ash borer that is basically causing the extinction of ashes, which are planted tremendously in cities. In Ohio, I gave a talk to a university in which they calculated they had 15,000 American ash trees on campus. And they were all dying. And I said, well, here's a golden opportunity to replant and actually have an experiment. And it seems to me we have so much restoration going on in the United States and around the world, in which we have no idea where these trees are coming from, the genetic variants or the source populations. And so by, I think every restoration project should be a genetics experiment in which we can learn this. And I think it's so important that it should become part of law of tagging plants in every restoration planning, whether it's in a city, around a courthouse, or in somebody's yard. And we know exactly which trees on the landscape, where that came from. And so then throughout Europe, I know you have, you still have natural areas. And so those could be the sources of stock that get planted and reciprocally planted in different environments. So I think this would be very tractable in Europe, just as it is in the United States. And then I think the velocity of climate change or global change is also a really important issue. Just how fast can we expect plants evolution and community evolution to occur? So I forgot to mention that example of the introduced elk. Those elk exclosures, some of them had only been up 7 years. And yet in just 7 years, we saw a genetic shift in, say, in the flowering time of the goldenrod. And I know that in annual plants, brassica in California, just a few years has been enough to switch their flowering phenologies as well. And so we're not necessarily talking hundreds of years here. I think a lot of evolution is going to happen really fast. And with the pinyon pine example, those switch in drought tolerant and drought intolerant genotypes. That was just 1 year. And that was based upon the standing genetic variation on the landscape. But of course, once you exceed the standing genetic variation, then you're going to be dependent upon mutations. And of course, that could be a much longer process. But actually working in the nursery industry, when you have thousands of plants in a nursery, one of my jobs as a kid was to walk the line looking for somatic mutations. And actually somatic mutations occur spontaneously and with surprising frequency. And just within a nursery, looking at somatic mutations for functional traits, we found variation occurring with just, within a few years. And so I think we think a lot about the evolutionary process as being so long, but I think climate change is going to result in our documenting pretty fast evolution because the evolutionary pressures are, well, you saw the sea of death for both the pinyon pine and the cottonwoods. That's pretty hard selection. And so I think we're going to see it. And this is a huge opportunity to understand evolutionary processes that would have been much harder to detect prior to the significant climate change that I think it's going to be far more obvious. A long answer, I'm sorry for that.
No, that's great. Thank you very much. Any others? Could I possibly ask you, you hinted at the beginning that there is a bit of controversy still about the idea of community selection. And I wondered if you might expand a bit on whether or not, do you still feel that there is controversy? I mean, I guess we're talking about what the unit of selection is here, whether or not it's individual or can be acting at the community level? Is that still argued, would you say, in a way that
Oh,
still
yes. It's interesting that we get far less pushback from quantitative geneticists. Quantitative geneticists immediately appreciate that relying on Mendelian genetics is kind of a no-go. I mean, there's so much quantitative genetics in agriculture that, you know, they're far more accepting. And there's lots of examples of selection and agriculture based upon group selection, family selection. And so the pushback, again, among geneticists is far, far less than I find with ecologists. And so I kind of find it interesting that the group that knows the least genetics is doing the most pushback. That shouldn't necessarily be surprising, but nevertheless, and so we've had, fortunately, in our group, we have a lot of geneticists, bona fide geneticists, in all of these areas, and so they have been, well, they've been part of the process, and so we've worked really hard to merge our disciplines, and I think that's another thing that's going to become increasingly important, is getting all of these different disciplines together. And so probably one of the hardest areas to merge has been ecosystem scientists and molecular geneticists, because a molecular geneticist finds little common ground with ecosystem scientists, and the vice versa is true. I mean, they talk different languages and they talk at different scales, but by having them all at the same table, we've realized that the term, for example, of an indirect genetic effect is very different to an ecologist and a geneticist. And so from the get-go, they were using the same words, but they had very different meanings. And we realized that they were talking at such cross purposes. And finally, we said, hey, these words mean different things. And so we need to be on the same page here. And once they started really talking, they realized that there was some really common. And so once you have a genetic basis to an ecosystem phenotype, like nutrient cycling, the ecosystem scientists could immediately see there was a connection to a genetic base. Once there's a genetic basis to nutrient cycling, then they were on the same page and had common ground. And so I think we just need to train a new generation that you don't have to actually be functional in all these disciplines, but at least you need to have an appreciation of what the other guy's doing. And I think, you know, like Jackie and Hillary, they've been exposed to this throughout their whole careers or graduate careers. And so I think to them, it's just the way you do business. But I think we still have a long way to go because a lot of groups do not communicate across levels. And I think that results in things happening much slower. But I think EVOLTREE, for example, is, you know, a great opportunity to merge all of this stuff.
Well, I fully agree, and I think on that point, that may be quite a good moment to draw us to a close. In fact, I think we're running near to the end of our time, and it's been yeah, really great to hear from you. I guess I could just allow maybe one other, if anybody has a late question. If not, then I would just like to thank you, Tom, and Jackie and Hillary, for agreeing to work with our time zones and to put together some content for us. It was really great to hear from you, and yeah, really enjoyed the talk and the discussion afterwards. So thank you very much indeed.
Well, thank you very much. And I participated in some other EVOLTREE conferences before, so I'm familiar with the group. And if you have any questions, Hillary, Jackie, and I would be happy to entertain any questions you have. Just email us and we'll do our best to get back to you.
Great. Thank you very much indeed.
Thank you.
Great. Well, thanks everyone. Just before you go, I think there's one final comment, one note. Do you have a slide, Christian, to share? Just to let you all know that the next EVOLTREE conference will be coming up next year in September. So mark the dates, September 12th to 15th. There will be details appearing soon on the EVOLTREE website, so do look out for those and please come along. Otherwise, it just remains to say thanks everyone for coming along. Thanks again to our speakers, and do come to the next one, which will be on the 30th of November. See you then.